discounts for icon passes. So if you ski or snowboard and you want a really good discount, you can check out our link in our Instagram bio at CSU Freeride. We also do payment plans for those icon passes. So if you pay 25 bucks now, you can slowly pay it off so you don't have to pay the entire thing at once. We're also going to have a table in the plaza on Thursday, so if you want to come out, meet some really cool people, and just hang with us, get to know us, we'd love to meet you all. So yeah, thanks for your time.
Thank you. All right, how are we doing? Is the microphone okay? All right, it has been a long time since I've been in front of a room this big. I'm used to projecting, I'm not used to having microphones, so if I start screaming and like blowing your eardrums out, just let me know. Just like wave me down. For those of you who don't know, my name is Michelle Steros. I'm going to be teaching this latter portion of Unit 1. And then Dr. Walren will be back for neuroscience. There's no way he's ever giving up neuroscience, so you can be assured he will be back. Until then, we'll just finish out where we started with the cell. And so we're going to go through, finish talking about the nucleus, where the nucleus in a eukaryotic cell was important for our DNA storage. Remember always that DNA stays in the nucleus, it does not leave. We're going to organize that DNA into chromatids on histones. So we'll talk about how we can wind that and make it nice and neat, but how that's going to cause some problems later when we want to read that DNA. We're going to talk about how we do move some things out of nuclear pores, not DNA, but we do have to move RNA out so that we can make proteins from it. And then we'll talk about how we can make proteins at the endoplasmic reticulum, the difference between smooth and rough ER. So smooth ER is something we're not going to get into. We don't really discuss. It is a part of the ER that does not have ribosomes studded. So it's smooth, hence the name. And it makes lipids. So like I said, we're really not going to talk about lipid synthesis beyond what I just said there. So that's pretty much smooth ER for you. You're done. Endoplasmic reticulum, rough endoplasmic reticulum is where we're going to spend most of our time. That's important for protein synthesis, specifically for these membrane-bound proteins. The transmembrane, meaning that you're plugging this into a membrane. Secreted, meaning that you're actually putting this out into the extracellular space. Or lysosomal, which is stuff that's going to go to our garbage disposal. We also have free ribosomes. Those are ribosomes that are functioning just in the cytoplasm. They're not attached to the endoplasmic reticulum. Those are going to be important for doing cytoplasmic proteins, proteins that we're going to keep in the cell and use within that cell. All right. And then finally, the ER is really important for glycosylation of proteins, adding sugars. Whenever you see glyco or gluco, think sugar. So this is a covalently linked sugar to all of those transmembrane lysosomal proteins. Once we're done with that, we have to transport it from the ER to the Golgi apparatus. Oh, I knew what I forgot. Sorry. Okay, so we're going to transport from the endoplasmic reticulum to the Golgi apparatus, and this is going to be additional processing. I like to think of this as the UPS store. You've made your protein, you send it off, now it has to get packaged, and it has to have a label put on it so it can get sent to the appropriate place. Once you've done that, you can send it off either to the plasma membrane if it's a transmembrane or a secreted protein, or to the lysosome if it's lysosomal. And then that's going to lead us into a deeper discussion of endo and exocytosis, if I actually manage to get there. So I had every intention of coming in here and drawing before we started, because this is going to take me a minute. And I'll show you pictures too, because I'm not the best drawer. So we'll be able to get a better look at this. But this is a cell with a nucleus, an endoplasmic reticulum. You're going to have Golgi apparatus. You're going to have lysosomes. I should probably label all these. Okay, on the nucleus, we're going to have these nuclear pores. We're also going to have ribosomes on some of these. And it's unlikely that we'll get to it today, but we'll also talk about mitochondria. I need more colors. Okay. Okay, so this is just going to be a reference. We're going to keep coming back to this kind of our big picture. So let's start talking specifically about our nucleus. So typically when you guys see pictures of DNA, you'll see it all nice and neatly arranged as the individual chromosomes. Of course, when we show you pictures of DNA, like just DNA, you always see those alpha helical structures. So how do we get from those nice little pretty alpha helixes to these very badly drawn chromosomes in chromatid form? The other thing to keep in mind is that, and these are numbers, please don't ever memorize numbers unless I tell you you specifically need these numbers, but just to give you an appreciation for this, if you were to lay out all of your chromosomes in a row, you would have about, I think it's a meter. And if you're thinking this is America, please speak English, that's about 3 feet. Okay. Sorry, I have trouble sometimes, even though we should be using metric for science. The thing is that it's going into this nucleus that's a lot smaller. The nucleus can vary depending on the particular cell type, but it's typically something on the average of 10 micrometers. Okay, that's like 10 to the negative 6 meters. In other words, you're shoving something into a container that's about 100,000 times smaller. So we have to have some way of really keeping this nicely contained and wrapped. And we do that via the histones. Wow. Okay, so histones. These are proteins. You could think of them as little spools. But they're going to assemble into octamers. Octo meaning 8. So you could actually put 4 on the board here, and then you would have 4 behind that. Once you have this octamer set, now you can start wrapping one of your chromosomes around it. So if this is your chromosome, I'm really not good with 3D, but you just wrap it around. You're not going to get your entire chromosome on one octamer. You're going to have multiple. So I'm not going to draw them all, but we'll draw one more set. Just do that. Okay, that's all well and good. Now you've got it wrapped up a little bit, but it's going to go even further. These histones now are going to wrap themselves. So we call that coiling. Okay, and as it coils, it can actually form into the shape that we've seen before. But as I said, this leads to a problem. If we have this nicely tucked into these chromatid forms, how do we get to the DNA that we need? Every one of these chromosomes is going to have genes on it that we need to turn on and transcribe and make proteins from. So say you had this gene that's right here between those pieces. It's not only wrapped around that one octamer, those octamers are now coiled. So you have to unwind to get to it. We're not going to get into the details of that. That's more genetics, cell bio. But I do want to make one point. Again, it's a detail. We're not going to test you on it, but it's just really fascinating. You can have the exact same DNA as someone else. You could be an identical twin, but you will still have differences: physical, behavioral, your risk factors for diseases. Okay, so you can have two twins, both have a BRCA2 gene. For those of you who don't know, BRCA2 gene increases risk of breast cancer and ovarian cancer. One of those twins could end up getting breast cancer and one could not. It's because of environmental factors, something that we call epigenetics. And there's a lot of things that influence these epigenetics and whether a gene turns on or how often it's transcribed. But one of them is the histones themselves. Okay, so we can actually methylate this process. Let me do red. When we say methyl, that's a CH3 group. Okay, so you've got a carbon and 3 hydrogens. You stick that on something, you just methylated it. Okay, it's a way of signaling. So if we methylate these octamers, that could be a signal that this portion gets unwound more often and gets transcribed more often, that we turn those genes on. So it's a really interesting field, because if we can figure out how to control those epigenetic factors, we could potentially reduce the risk of certain genes turning on or increase good ones turning on. So, okay. Now we've wound it up, and then we had to turn around and unwind it so that we could actually read it and make RNA off of it. So I know you guys talked about RNA. I'm not going to go over it entirely, but just to review. We had mRNA. This is messenger. This is your blueprint. This is where you read the bases of the DNA and you match the RNA bases to it. You have to have something that's going to read that mRNA, and that's going to be RNA. Put that with some proteins and you have a ribosome. So the ribosome will read that mRNA, decide what amino acids are matching those bases in that RNA, and then you're going to have transfer RNA bring the bases, or bring the amino acids in to make your new protein. So that's your last one. And we'll talk about that in more detail and go through the mechanics of the entire process next week. But for now, we've had our DNA in here. We made some RNA off of it. What color do we want RNA? And now we can move it out, because we always do protein synthesis starting in the cytoplasm. We may move it to the membrane, the endoplasmic reticulum, but it will not be in the nucleus that we're making proteins. So it has to go out through the nuclear pore. One misconception I often see with students is that when we talk about these endoplasmic reticulum being surrounding the nucleus itself and then these nuclear pores, they get the impression that when you push something out the nuclear pore, you push it into the endoplasmic reticulum, which isn't true. If you, let me find a black. So I'm going to hash what we would call the lumen of the endoplasmic reticulum. So I don't know if you've talked about lumen before, but a lumen is just the inside of any organelle or organ. Okay. When we push something out of the nuclear pore here, we're actually going to be pushing it between the endoplasmic reticulum. It's a labyrinth, and I'll show you a picture where you can really see how complicated it is, but it will not be into the lumen of the ER when you start. Okay. I really wish I'd put that on the top board. Okay. So we're going to go from this semi-artistic version of an endoplasmic reticulum, and now I'm going to show you endoplasmic reticulum as a blob. So just use your imagination. Now, what color did I do RNA? So you had RNA. It got pushed out of the nuclear pore, and it's in the cytoplasm. It's surrounded by all this endoplasmic reticulum. This RNA is first going to get picked up by a ribosome. And it's going to start reading. And it's going to bring in a tRNA, and it's going to start making this protein. And if it turns out that this is an mRNA that's coding for a cytoplasmic protein, something that's going to stay in the cell, no problem. It can continue making it right here in the cytoplasm. It'll push out this protein. And you're good. But if this is going to be a transmembrane, a secreted, or a lysosomal protein, you can't just make it in the cytoplasm. You'd have no way to get it to where it needs to go. It has to go through the factory system. It has to go through the ER, get packaged in the Golgi, and then get sent to the appropriate place. So if this happens to be green, we'll do green. If there's a signal on this budding protein that tells it that it is transmembrane, secreted, or lysosomal, that's going to direct this ribosome to move to the endoplasmic reticulum. Yes?
The signal saying whether or not the ribosome is transmembrane, secreted, or lysosomal, or the RNA? The ribosome?
No, so the question was, is the signal on the ribosome itself or the protein? And it's the new protein that's being made. And we'll talk about the signal peptide next week again. But basically what it is if you have a series of hydrophobic amino acids, then that's a signal that this should be one of these three, and it will take it to the endoplasmic reticulum.
So the signals are on the protein that tells whether or not the protein is cytoplasmic?
Yes, the protein kind of gives itself away and says, hey, I don't belong here. Hold on just a second. Yes?
Black squiggle, is that the protein?
This is supposed to be a protein, yes. Cytoplasmic protein. Yes?
Okay, just so I'm understanding correctly, so the ribosome picks up the RNA, it reads it. If it's cytoplasmic protein, it stays in the cytoplasm and starts making it. But if it's not and it gets a signal, the ribosome takes it to the ER.
Correct.
Okay.
So what we're saying is if it's a cytoplasmic protein, if it does not have a signal on the protein itself, it will continue making it in the cytoplasm and release it into the cytoplasm. But if there is a signal on this protein that it's actually supposed to be one of these, then this entire ribosome, the mRNA that it's reading, and the protein that's been made is going to move over. So you'll actually have your RNA piece that it's reading, and this protein that you were making at the ER now. I think you were next.
So I know that the mRNA, or the RNA and the ribosome find each other just basically through diffusion. They kind of run into each other. But how does the ribosome get to the endoplasmic reticulum?
That's a great question. She wants to know, how does the ribosome get to the endoplasmic reticulum? Starting tomorrow, we're going to start talking about cytoskeletal proteins that act as highways in this cell, and the motors that run on them. And so we'll talk about things like how we're going to move things around the cell. Great question. Yes?
Is this happening during translation, or?
This is translation, yes. So this is kind of a preview of translation when we're reading RNA and making a protein from it. Yes?
So the signal is it already the signal is attached to, or is it formed during translation? Or?
The signal is part of the protein itself. It's the amino acids within the protein that say, I don't belong here. Yeah. Yes.
Okay, so is the protein finished being created, or is it kind of like starting, like it's reading it and then it gets the signal and then it's like, okay, we're going to finish creating this protein in the ER? Or is the protein created outside of the ER when it's like one of the transmembrane secreted system, and then it's like, oh, now we got to send it to the ER?
So if it has that signal, if those first proteins are going to be too hydrophobic to be in this area, it will stop translation to move to the endoplasmic reticulum and then finish there.
Okay.
Yes.
Sorry, I'm a little confused. Is RNA reading the protein?
So this is the RNA that moved out. We can read here, this is mRNA. That's what you're reading. This ribosome is really our RNA. Reading the protein. Oh, there's a whole, we're going to talk about translation in detail, and we will go through every single step of this and all of the different proteins that are involved, including what will read that signal. But I just kind of wanted to give you a preview now.
Oh, okay.
Yeah, so don't get too caught up in the details because we will go over this very well. Yes?
So it's reading the mRNA,
Mm-hmm.
turning that into rRNA?
No, the rRNA is going to make up the ribosome. That's what a ribosome is. It's our RNA. And it's the ribosome that reads the mRNA, and then it makes a protein. So that's the idea of the central dogma, DNA to RNA to protein.
What about the tRNA?
tRNA, we'll talk about how tRNA is involved in the process next week. tRNA is going to actually bring those amino acids to the ribosome to add them to the protein. Yeah.
Does the ribosome just go to the edge of the ER, or does it go
The ribosome will actually bind. Okay, so when you look at them, you'll see it kind of looks like they're studded in. It's not, there's another player in here that's going to actually bind and hold that protein. We'll talk about next week too. Sorry guys, I thought we would just do a quick review of RNA, but that led to some confusion. So we will get that straightened out next week, I promise. We'll spend several days talking about all of this. Yes?
So is this then the rough ER where it's binding, or is this just completely different than that?
Exactly. This is specifically, if we're binding, this has to be rough ER. Because the smooth ER is only going to do lipid synthesis. Yes?
So,
Go ahead.
so once the RNA leaves the nucleus through the nuclear pores, it's just kind of inside plasma, and that's where it starts being translated from, the RNA starts being translated by ribosome, and then depending on what signals on the protein, it's either going to be cytoplasmic, or that whole complex is going to be shifted to endoplasmic reticulum if it's transmitting very superior lysosomal signal.
Exactly. Yes. I'm going to try to get this picture up because I think this might help a little. What the heck is that? Please work. There we go. Thank you. Okay, can you guys see okay? Do I need to turn any lights down? All right. I don't have a functional pointer, so I'll just be like hand waving. But you could see your double helix formation, and then you could see the individual histones that are assembling into that octamer, those 8 together. And then the DNA is going to wind around it, and then from that, those octamers can actually coil and supercoil into your chromatid. Yes?
And methylation winds it tighter so it becomes unable to unwind? Or?
I think methylation is a signal to unwind,
Oh,
but I
signal to
could,
unwind.
yeah, I'm not 100% certain. It could be that it does both. Okay, here's a great picture. So all the squiggly stuff in the center is your nucleus. All the stuff that you could see colored green, that's the outside of the endoplasmic reticulum. The dots you see all over that, those are the ribosomes studding it, so that tells you that's rough endoplasmic reticulum. And then the kind of tan portion, that's the lumen of the ER. So you're going to make your RNA, you're going to pass it out through a pore, and there's going to be ribosomes just all over the place there. We'll call them free ribosomes because they're just in the cytoplasm. They'll pick up the mRNA that comes out and start reading it, start making a protein. If it's cytoplasm, it'll just finish. If it's a transmembrane secreted or lysosomal, it will move the entire complex to that green portion, and it will stud in and feed the protein into the endoplasmic reticulum. Does that help a little? Okay, good.
Does it attach to a new ribosome in there, or does it stay onto that free ribosome that was already attached to?
It's going to stay on. When we say free and membrane-bound ribosome, we're just talking about location. So once we stud a ribosome onto the endoplasmic reticulum, it's a membrane-bound ribosome.
Oh,
Okay. So typically we use those terms. We'll say membrane-bound ribosome just to kind of tell you what kind of protein it's making, because you know the location. Yes? I'm sorry? What does what mean? Lumen. Lumen is the inside of anything. So the inside of an organelle or the inside of an organ. So, I mean, these are actually membrane-bound compartments. If you were to look at it like this, anything inside that membrane is considered a lumen. Yeah.
So would these ribosomes pop on, then become membrane bound, and then when they're done, pop up and become free again? Or is it just one-time thing, they're pop on, they're on?
Yeah, we don't talk about that, but yes, they just return to being free. And then they'll pick up another mRNA and decide whether they can stay there or they have to traffic them over. Yeah. All right. Let's talk about what happens when we start pushing things into the ER. I'm going to do it again. Okay, so we've got our rough endoplasmic reticulum. We've got a ribosome, membrane bound. We're going to start reading mRNA. And we're going to make our proteins. We're literally going to feed these across the rough endoplasmic reticulum membrane. If this is going to be a secreted protein, sorry, it will be completely pushed across the membrane and into the lumen of the ER. If it's a lysosomal protein, green, same thing. But if we're making a transmembrane protein, a protein that's meant to have those passes across the plasma membrane, we're actually going to start threading it into those passes here at the ER. So we will make it. I'm going to do, well, we'll still do black. And it's going to thread. Okay.
Where does it, sorry,
Go ahead.
does it thread? Like, what do you mean by thread?
Thread meaning it's going to pass it through the membrane back and forth. So each pass will go through. So this would be 1 pass, 2 pass, 3 pass.
Does that mean that it's going to pass into lumen and back out and then pass the lumen back out again?
Exactly. When you make this protein and you're pushing it through, you're going to push it from the extracellular side to the lumen, and then it will go back out to the extracellular space into the cytoplasm, not extracellular space, I'm sorry, into the cytoplasm, and then back to the lumen again. Again, this is something that we're going to talk about in detail when we talk about translation. We'll talk about the mechanism of how we actually thread this. Yes?
And so the transmembrane protein, is that made from secreted protein or the lysosomal?
No. So these are 3 different types of proteins you can make. So I could show.
Membrane, that's when it would go back and forth through the membrane.
Yes. So you can actually just push an entire protein through, and it would either be secreted or lysosomal if it's in the lumen, or you can make one that's going to thread. But they're going to be completely different. If it threads, it's transmembrane. So the last thing we need to talk about the ER being responsible for is doing that glycosylation, doing those sugars linkages. Okay, so we're going to link these sugars to our proteins. And these sugars can serve a lot of purposes. They can be things like signals. They can work as barriers. I always like to think of them kind of as address labels. They do work to some extent in telling the cell where it needs to go. But one thing I want to show you is that what we're using to put this on, another protein, a transferase, to put these sugars on, it works in the lumen of the ER. So if you have secreted or lysosomal proteins, you can have sugars all around it. But if you're making a transmembrane protein, you can only glycosylate the portions of that transmembrane protein that are in the lumen. The stuff that's facing the cytoplasm will not be glycosylated.
Just to clarify, so the sugar is kind of like a signal for the function of the protein?
The sugars have a lot of different functions, but oftentimes they act as signals. Could be recognition, could be to target it to someplace. Yes. Okay, so now we've finalized our proteins. They're made, they've got sugar on them. Now we need to move them to the next step. Okay, so we've made our little proteins in here. And we've got to get them to the Golgi apparatus. Now, the Golgi apparatus is made up of a bunch of stacks, typically probably 10. I'm just going to draw 3 for simplicity. We say that the ones that are closest to the nucleus are cis, and the ones closest to the membrane are going to be trans. So to move these proteins, we need a vesicle. And a vesicle is just a part of a plasma membrane. It's just a small portion. We can make vesicles by just blebbing off a portion of whatever membrane we're working with. And then we can reabsorb them when we get to another membrane. Remember, these are phospholipid bilayers, so they all have the same components. It's no problem to kind of move them back and forth between different membrane-bound organelles. So if you were to start blebbing off, and I'm going to move this. By bleb, I mean you have this kind of start sticking out a little bit. And you're going to shove all your proteins in there. The transmembrane protein is already in the membrane itself, so you just bleb off that portion. You shove the lysosomal and secreted in there as well. And then this neck will just get smaller and smaller until it comes off. Okay, this is now going to travel to the cis portion, the most cis portion of your Golgi apparatus. And merge. So again, once the vesicle has merged with the Golgi apparatus membrane, you're going to carry over this transmembrane protein. It's now just part of the Golgi membrane. And you're going to have your secreted and your lysosomal proteins in the lumen of the Golgi. Okay, in the Golgi, we're going to do things like add more sugars. So it already had sugars. I kind of quit drawing them. But now we're going to add Golgi sugars. One point I want to make about these glycosylations. Glycosylation. If you're in the endoplasmic reticulum, the type of sugars you're going to be putting on, or I should say more where you're binding them, it's going to be, oh great, N-linked sugars. You do not have to understand what that means. We're not going to ask you to know anything about the actual chemistry or the chemical bonds there. Just recognize that when you see an N-linked sugar, that occurred in the endoplasmic reticulum. And I always remember it because it matches the second letter. So now if you were doing Golgi apparatus, the second letter is O. Those are O-linked sugars. And I really probably should have made them match the sugars I put on there, but yes.
Okay, so I kind of lost a little bit. So we do a merge onto the Golgi apparatus. Then what happens to the proteins? Does the stay on the outside of the Golgi apparatus?
Yes, this is your transmembrane protein there. So it will stay in the membrane of that Golgi apparatus. And then your lysosomal and your secreted proteins will be in the lumen.
So the transmembrane protein stays in the membrane of the Golgi apparatus?
Yes, once you make a transmembrane protein, it always has to stay in that membrane. Remember when we talked about those passes, we said that it's going to be in contact with those fatty acid tails. So it's going to have hydrophobic R groups. You can't put that in a lumen in a hydrophilic environment.
Were the proteins supposed to be glycosylated while they were in the vesicle as well?
Yes, I got lazy, sorry.
No. So when the proteins enter the Golgi apparatus, that's when the Golgi apparatus adds the O-linked sugars?
Yes.
And the Golgi sugars are added?
So these are the O-linked sugars are in blue. They got added here in the Golgi. And then the red ones were the N-linked sugars that were put on in the endoplasmic reticulum. Yes?
So with the Golgi O-linked sugars, are those only added to the inside of the transmembrane protein like the N-linked sugars?
Yes, this glycosylation pattern in the Golgi apparatus is going to follow the same rules as the endoplasmic reticulum. That means that if you're in the lumen, you can glycosylate the entire protein. But those transmembrane proteins will only have the lumen portions that can be glycosylated. Yes?
So in the Golgi apparatus, it has both types of sugars, N-linked and O-linked?
Yes. So both the N-linked and the O-linked sugars here. Okay, it doesn't lose any of this glycosylation. It will keep it all.
So then with the transmembrane protein in the Golgi apparatus, does that just mean that the parts of the transmembrane protein that are in the lumen of the Golgi apparatus, those are the only ones that can have the O-linked sugars?
So when you're talking about these transmembrane proteins, you're going to have the N-linked, and then you're also going to add on the O-linked, but it will always only be on the side that faces the lumen.
Stick together with each other, or there's like gaps?
There's typically a bit of a gap between most of these. It's a pretty complex structure, and there's connections between them, but there's gaps as well. Yeah. Okay, so now you've done what you need to do in this portion of your Golgi. You just have to keep moving it through the stacks. And it will continually get processed. We're not going to get into the details of how it's processed, just that it keeps passing. And when it does that, it's going to do the same exact thing. It's going to bleb off a portion of its membrane. I'm just going to kind of draw like a blub here. And then that's going to merge on the next one. So it's going to take all those, it's going to dump them into the next one. When it's done there, it'll blub them out again and pass them to the next one, all the way through to the most trans portion.
So because the Golgi is layered like that, it doesn't require vesicles of those proteins, so will they just go down from one to the other?
It has to move through vesicles from one stack to the other. So this bleb here will become an actual vesicle that then merges and dumps it into the next stack. And then each way through, yes. Okay, and then you get to the most trans portion. Go ahead.
I was just gonna ask why does it have to go through all of the Golgi bubbles?
Each stack, I think, has a slightly different process of modification that has to occur. So you could think of it like a factory. You've got one portion that's going to, screw this down, and another portion that sticks this sticker on it, and then this side paints it. It just goes through a whole process of modifications. Here's where things get different. Now you're down here. You have this transmembrane protein. And then you have your secreted protein and your lysosomal. Now you don't just want to grab them all into one vesicle, because these are going to different places. You've got your transmembrane and your secreted both going to the plasma membrane, but your lysosomal needs to go over to the lysosome instead. So this trans portion will have proteins that recognize where it needs to go, and it will make two separate vesicles. So you'll have the one vesicle that's going to have your transmembrane and your secreted. I should probably redraw that because it's okay, so that is going to actually bleb off. It's going to become a vesicle, and then it can merge with that plasma membrane. Whereas you're going to have a completely different vesicle here with your lysosomal protein, which is still glycosylated. I'm just not always drawing these.
So the secreted protein can merge with the plasma membrane?
Yes. This vesicle, once it leaves the trans portion of the Golgi apparatus, will be able to merge with the plasma membrane. And the secreted and the transmembrane protein will be in there. And I will show that to you. I'll draw that out on a different board.
For the vesicle that's merging with the plasma membrane, does that vesicle contain the transmembrane protein and the secreted protein, or are they two separate vesicles that merge with the
No, they could be in the same vesicle for the transmembrane and secreted, because they're going to the same location. Yeah.
So I think you just answered this, but I'm going to clarify. So secreted and transmembrane go in the same vesicle, and then lysosomal goes into a separate vesicle?
Correct. So I'm going to redraw it here so that we can actually see this. So this is going to be the most trans portion of our Golgi apparatus. We're going to have that transmembrane protein in here, the secreted, and our lysosomal. Oops. And from this, we can get 2 separate vesicles that are going to bleb out from this Golgi apparatus. You'll have this one vesicle, not to scale, it's almost as big as the Golgi, but none of this is actually to scale, your cell's much smaller. This is your transmembrane. This is your secreted. Here's that plasma membrane. So this is going to come to this membrane and it's going to merge. So this is now your extracellular space. So when you actually take that vesicle and you merge it into the membrane, it will become part of the membrane and flatten out. What's in the vesicle can exocytose, exit the cell, and it's secreted into that extracellular space. The transmembrane protein will just stay exactly where it was, and now it's just part of this plasma membrane. Notice here that because we could only glycosylate the luminal side in vesicles and lumens, that when we actually put this into the plasma membrane, the sugars are going to be facing the extracellular side. And that makes sense, hopefully, because you don't really need to be signaling back within the cell and trying to recognize in the cell, you're recognizing things in your extracellular space. Okay. The other one that we can create here, we're going to have the second vesicle that comes out. This had our lysosomal protein. What color did I have lysosomes? Red. Okay, so here's your cell. And you've got a lysosome right over here. This vesicle can merge with the lysosome. And now you have that lysosomal protein delivered to the lysosome. Okay. All right, I'd hope to get a little further, but I think that's probably the best place to cut off, and we'll start again tomorrow.